Thank you for the invitation to come here and for lots of fascinating talks this morning. And I'm going to sort of talk about why some of the evolutionary thinking about diseases has actually not progressed so far. When we're talking about a whole class of diseases, those are diseases caused by pathogens, right? And so what we've had is a wonderful set of talks about when the body doesn't work properly in a whole variety of situations which are relatively sort of beginning to get amenable. They're beginning to get amenable because we understand a little bit about the underlying sort of physiological system which we're talking about. So we understand a little bit about the digestive system, the respiratory system, etc., etc. A conjecture is we haven't done that much for, even though it's a huge topic in public health, of why pathogens actually cause disease and really understanding that. And I think one of the reasons is that we've been really poor about understanding the immune system. And so really what I'm going to do in this talk is go a step back and ask, why do we understand so little about the immune system, and address that from an evolutionary perspective. Okay. So in other words, and I've stole this slide from Carl, sort of, you see, and this is a very simplified slide, that there are lots of complicated players in the immune system. And in spite of perhaps more effort being thrown by the NIH at the immune system than most other physiological systems, we still don't understand it. And it's not just complex, it's sort of perplexing. And let me just illustrate why it's perplexing with a very simple example. So we know that when we see a pathogen, or in this case a mouse sees a listeria infection, we can look at what happens. And the basic principle is very simple, or at least supposed to be very simple. So on the y-axis is the amount of listeria. This is as a time after infection, and this is coming from Eric Paymer's lab. And you see that the pathogen grows and then it gets cleared in about a week. This is a typical course of what's the simplest sort of infection we can possibly talk about. It's an acute infection. And the mouse is then immune to this listeria forever. Okay, or at least for as long as the mouse lives. On the right-hand panel, we are seeing the immune response as a function of time. And this is, this is what my colleague Bruce Levin calls immunoyak, sort of something complicated to make sure, but it's just the magnitude of the immune response in some way. And really, the basic principle which we thought we knew about completely was the principle of clonal selection. In other words, that there's very small responses to this listeria, essentially undetectable at the outset, and this response grows as it sees the pathogen. As the immune cells see the pathogen or notice the pathogens there, they divide, and this response grows exponentially as a consequence, and then gets maintained at some level forever. And so we thought we understood this. And then Eric's group, Paymer's group, did a very simple experiment. They treated the mouse with antibiotics at day 1, so over here. And of course, the result was not expected. So the pathogen got cleared right away. They gave the mouse enough antibiotic. It eliminated the bacteria. But surprisingly enough, the immune response sort of didn't know about this, didn't know about that it was supposed to stop doing that. It just kept on going as before. So it's clear that the immune system is not just complex, but the rules that are underlying it are not very simple. Okay. And so one of the reasons why, and one conjecture of why that could be the case, is that really the immune system differs from other physiological systems because it deals with a slightly different scenario. Other physiological systems, like the digestive system and the respiratory system, have to deal with variability. We eat different sorts of food, at least lots of us like trying different sorts of food, particularly when we go to different meetings. We go into areas which are different with regard to temperature and humidity and so for our respiratory system. But that's relatively random heterogeneity. The immune system has a fundamentally different problem. And potentially the difficulty arises because the immune system is dealing with something, the pathogen, that's trying to evolve to escape it. So it's almost as if the food's trying to escape our digestive system, the air's trying not to be breathed. So the basic principles of the immune system are likely to be different. Okay. So about Carl and I have been talking about this for a long time, but about 5 years ago I sort of took a year's sabbatical in Seattle, and we spent some more time talking about it, and some of these ideas came from there. So the question is, the immune system is now in an evolutionary sort of arms race with the pathogen. And what we're interested in is understanding how the immune system can deal with this evolutionary arms race when the pathogen has huge population sizes and much faster evolutionary rates than the host has. So how can it do that? And what I'm just going to catalog is a little bit of what we came up with. Well, the first thing is, if you want to do that, the immune system needs to evolve. And really, we know that. The adaptive immune system is really an evolutionary system that's working within the host. And that's really what the principle of clonal selection in the immune system is all about. The immune system generates this diversity right at the beginning, and we all have this diversity. An interesting problem is we actually don't know how big this diversity is. Even though immunologists have been measuring this for years, together with Joe Blattman, who's actually in the immunology program over here at Arizona, we're trying to estimate this. Because for technical reasons, no one's been able to really do that. So it generates all this diversity, and then it's really evolution within the host. The pathogen selects for the expansion of the useful and sort of ignores the other immune cells, which are not pathogen specific. So a given red pathogen will select for the expansion of just the cells that can fight that pathogen, and then some will be kept around for a long period of time. So really, the evolutionary timescales aren't all that different. The immune system can evolve pretty much as fast as the pathogen. Okay? But then you might expect to see an ongoing arms race in almost every infection. You'd expect to see the pathogen, the immune system makes a response to it, the pathogen varies, the immune system will have to play a constant game of catch-up. It does happen rarely, and that's called antigenic variation, and it happens in trypanosomes. You see consequences of it in HIV, but it doesn't happen for most infections. So the question is why it doesn't happen. And there's sort of some very basic evolutionary reasons for that. The first is that the host actually selects for one sort of diversity, which is diversity at the MHC level. And what that does is really that each host, and all of us, at least so far, and especially in the United States thus far, are very diverse, right? And as a consequence, each of us sees the pathogen in different ways. So we sort of break up the pathogen and see it differently in each of us. So the pathogen really has a very hard time to adapt. And so really, and this is particularly true for a group of responses called T-cell responses, this prevents the pathogen really escaping. The other bit that the immune system does, which is very simple, is it makes lots of responses. So it makes responses to different bits of the pathogen, and that's very similar to the idea of multi-drug therapy, is if you make responses to lots of things, the pathogen has to move away from all of them to escape it. It can't do it very simply. Okay? And interestingly over here, the sort of part of the immune response to T cells which look at MHC, it turns out that pathogens can find it much more difficult to escape from them. So influenza doesn't actually manage to escape from T cells. It's another reason why they don't control influenza strain variation very much. But the antibodies do manage to escape, or at least, sorry, the virus manages to escape from antibodies. And antibodies in all of us recognize the same basic things. in fact, is a really curious sort of result that came out recently, is it turns out in lampreys, which have a completely different immune system from us, adaptive immune system, Max Cooper, who's at Emory, did a wonderful experiment. He said, what if I immunize a lamprey against flu? What will its immune system, which is completely different, recognize? And totally to everyone's surprise, it recognizes just the same thing as our immune system recognizes. It's fascinating. It's sort of irrelevant to this just now.
I was smoking when he came up with the idea.
Well, you want to know it because you want some of that, right? Right. Okay, so that's one way the immune system works. So another way the immune system could work, and that came out of work with Carl and Eric Chastain, was you want to sort of slow down, you want to force your competitor to evolve slowly. And this may seem a little perplexing, and I'll try and explain it just now. So you can think of it this way. The pathogen gets seen by the host, and the host has to process information. So it has a sensor to see the pathogen. It makes signals between all the different components of the immune system. It decides what to do with all those signals, integrate all those signals, and make a response that controls the pathogen. But the pathogen can do a bunch of stuff, which is quite nasty. It can think of how to tamper with these signals. And so the host's processing information, and the immune system has to do that, and the pathogens can tamper with this. And if I distill this into a nutshell, we can ask, how do pathogens prevent hosts, how do hosts prevent pathogens tampering with these signals? So one way to do that actually makes sense of this puzzle that I gave you early on. One way to do that is not to sort of integrate signals, not to look at what your enemy is doing all the time. What the immune system tends to do over here is that it's looking at the pathogen early on. It's looking at it for the first day or two after infection. It makes up its mind what to do, and then it ignores information after that. And actually, not using all the information can sometimes be better than using all the information. And in particular, you'd like to use information when it's likely not to be subverting you, which is when the density of the pathogen is relatively low. Pathogens sort of, their tampering mechanisms are usually generating cytokines and other molecules that the immune system uses. And when their density is low, they can't really tamper with these signals. When their density is high, they can, and so you just don't listen to them. Okay. There's another way of doing it. The other way of doing it is to notice that while the pathogen can evolve much faster potentially than the host, it's moving, at least in sort of the space of how the immune system works. It's moving, the host makes a static fitness landscape, and the pathogen's moving on it. So you might think that the pathogen really has an advantage in this game, but it's actually not necessarily the case. And really the key insight is the host has an advantage, is in it sets the game. It chooses the landscape. It's not that the pathogen and the host can both choose the landscape. It's that the host chooses the basic landscape, and then the pathogen's forced to evolve on that landscape itself. And so, as we know, and as all evolutionary biologists know, if you want to evolve slowly, or you want something to evolve slowly, choose a rugged landscape. So that essentially, it'll take a much higher time for the pathogen to reach the fitness peak. Okay? So that might be one of the reasons. And so, really, one of the hypotheses for why we have complex networks over here is because these complex networks might be the host's response to slowing the pathogen's evolution down by generating a rugged fitness landscape. this sort of explains this, that while these networks are strongly conserved, they haven't had to change. If really there'd been an ongoing arms race between the immune system of mice and pathogens versus men and pathogens, as mice and men diverged a long time ago, they would have relatively different immune systems. But no, pretty much all our immunology can be done in mice, and it translates to humans. Another bit is it shows that many of these cytokine networks show a lot of signs of purifying selection rather than positive selection, which has been puzzling. And it contrasts with stuff at the MHC where there's actually a reason to think that we want to be different. Okay. And this also, as Carl likes to sort of point out, explains the bit of why immunology is so complicated. And it's really because if pre-meds could understand it, pathogens could understand it too. Okay. And as is usual, we've sort of got some ideas of where to go, but we have lots of unsolved problems. And perhaps the two big unsolved problems are, one is, how do we evolve such a robust and complex and robust signaling system? That's the first one. How we get there is an unsolved problem. Another one is, how do we use this information to actually figure out how the immune system works? And I'm sort of going to summarize and end over here by saying that really, when we look at the immune system, which is designed to maximize fitness of the host in a hostile world, it does so by really generating diversity and evolving at the same time scale of the pathogen. And in order for that to work, it evolves to look at multiple targets and have different targets in different people. It also has to be robust against evolving pathogens, and really there's a little bit of how to look for signals and how to design signaling networks. There's other bits that we've been working on in this regard, is that if you have to do this, you have to sort of generate responses against pretty much anything the pathogen can put up. The problem is avoiding generating a response to yourself and really minimizing pathology. And some ongoing work we've been doing in the lab is understanding how regulation in the immune system works, and there's some very nice population biology possible there. And finally, something we've been doing, which I don't have time, but I'll be happy to talk with other people, is can we use evolutionary biology to actually help us design better ways to train our immune system and actually get information from how pathogens are trying to subvert them to improve the way we design vaccines. And that's some very recent work we're doing with Ralphie Ahmed's lab. Okay, so thank you.
This was a very interesting talk. Thank you for this. And this makes me think about tuberculosis, where people can be exposed to tuberculosis and be infected with it, but not have the disease. Yeah. And they don't get the disease of tuberculosis until their immune system fails or they go into immunosenescence. So why doesn't the host system recognize the bacillus and clear it out earlier?
Yeah. For TB, it's a wonderful puzzle, right? It's one of the cases we really just don't understand. We don't even know. We know that if you take out the immune system, people come down with tuberculosis. So the basic observation is, in a lot of the world where tuberculosis is prevalent, pretty much everyone's infected. But only about 10% of people progress to tuberculosis in their lifetime. We don't know why, right? We know that if you immunocompromise people, and HIV unfortunately does that, you get a much larger fraction progressing. But in natural circumstances, we don't know what breaks down, and it's not necessarily immunosenescence, although that's one hypothesis.